Hi all, welcome to Biology classes with Lakshmi Chandra. In today's session, we'll focus on some of the post-transcriptional modifications. So having completed transcription, let us see what are the modifications that happens after transcription. But you have to bear in mind that these modifications happens only in eukaryotes. These post-transcriptional modifications are absent in prokaryotes. Let's first examine why it is so. Why post-transcriptional modifications are present only in eukaryotes. Why only in eukaryotes? We can summarize the whole reason in 2 points. One is because in eukaryotes they have something called as the split genes. So presence of split genes in eukaryotes. The second reason is presence of nucleus. Fine. The second reason everyone knows, nucleus in eukaryotes, the basic feature that differentiates a eukaryote from a prokaryote. But the first point that I have stressed here, presence of split genes. So we should all know what is this split genes. Well, we know gene is a segment of DNA that encodes a protein, or a functional unit of DNA is referred to as a gene. This we all know. We have learned in the first set classes itself when I took the Mendelian genetics, there what gene is and what is genetics, such terms were well defined in that particular session. So here we all know what a gene is, but what is a split gene? Let's focus on that first. Split genes. I'm just going to draw a segment of DNA, just going to represent a segment of DNA like this, having 2 ends, 5' and 3' ends, segment of DNA I have drawn here. This DNA I have drawn, and this part that I'm marking it here inside, this is a gene here. That means this segment of DNA is going to express a protein. It has the information to express a protein. But then I am calling this gene as a split gene, because within this segment there are certain regions which have got the information to express itself, but there are regions which cannot express themselves. Okay, so within a gene there are regions which can express themselves and there are regions which cannot express themselves. So those regions which can express themselves means they have the information to produce an amino acid or to produce a protein. Okay, such regions which can express themselves or have got the information, they are called as the exons. Exons are the regions present in a gene which gets expressed. I hope you understood the meaning here. Expressed means it is having the information to produce a protein or to produce an amino acid. That is the meaning. Okay, so it gets expressed. So exons, I am just marking it as E. So this is my exon here, this region. This is another exon, this is another exon. 3 exons are there in this gene. But the other areas which are not marked anything here, they don't have the information to produce an amino acid. But they are there in between the exons, or they are interfering with the exons, or they are getting interrupted in between the exons. Okay, so such sequences which are intervening or getting in between the exons, such regions are called as the introns. Introns. Let me represent this intron by I, the letter I. So these introns, they are not getting expressed, but they are intervening. intervening, or I can say they are interfering. They're not getting expressed, but they are in between the exons. They're interfering with the exons. Okay, so such regions are called as introns. So this gene is having both exon as well as introns. Such a gene segment, you call it as a split gene. And this split gene is absent in prokaryotes. In prokaryotes, the segment of DNA or the gene which is coding for a polypeptide, the whole gene is having only exons. There are no introns present. But in eukaryotes, the gene has introns as well as exons. So since introns are also there, splitting up the gene, you call such a gene as a split gene. So that is one reason why we need modifications in eukaryotes. Post-transcriptional modification is required. Okay. Now, what is the relation between the split gene and the modification? Let me tell you that. So this is my split gene that I have drawn here. Okay. This split gene, this is DNA. Now we all know that during transcription, DNA is producing one RNA molecule. You call that as mRNA. mRNA is getting produced. So this mRNA which is getting produced will also have introns. These exons will be there in them. Introns also will be there in them. Exon will be there. Intron will be there. Exon likewise. So this mRNA which is produced will also have these introns and exons, that is sequence which are expressed and sequence which are not expressed. But then what is the purpose of this mRNA production? Why is this mRNA produced? It is produced so that this mRNA is ultimately producing a polypeptide or a protein. This is the basic function. This is what is needed here. So that is central dogma of molecular biology. Information is passed on from DNA to mRNA to proteins. So this process you call it as transcription, and after which there is a process called as translation. Explanation regarding translation will be given in the coming videos. So here so far we have completed transcription, the whole steps, initiation, elongation, termination, everything we have completed. So after that we are getting mRNA having intron as well as exon, but there is no need of this introns here, isn't it? We need only those portions which is able to produce a polypeptide. So in post-transcriptional modification, what is required is we have to remove all these introns. And then we have to join the exons. Exons are there, introns are removed and exons are joined. Only when the introns are removed and exons are joined, this can produce the actual polypeptide. Otherwise, the introns, the interfering sequences will be intervening with the exons. Okay, so removal of introns and adding of all the exons together is a post-transcriptional modification, and that is called as splicing. Splicing is removal of introns and joining of exons, and this complex process is done by the enzyme called as spliceosome. So now I think you could relate why there is a post-transcriptional modification in eukaryotes and how that modification is done. So why? Because it is having split gene. And how that is done? By the process of splicing. All the introns are removed and exons are joined using the enzyme spliceosome. Now dear students, dear viewers, one important thing you have to bear it in mind. This point here. DNA is producing mRNA, I told you. So this mRNA is having intron as well as exon, I told you. So this mRNA which is having intron and exon, you cannot call it as a true mRNA. Instead, you call it by the name hnRNA. Usually this is a mistake, or sometimes it goes out of our mind. We say DNA is producing mRNA. It's not like that. It is producing what is called as hnRNA. Now what is this HN? HN stands for heterogeneous nuclear RNA. So this is producing HN, heterogeneous nuclear RNA. So DNA is inside the nucleus, we know. This RNA which is produced is also inside the nucleus. So this is heterogeneous. Heterogeneous means it is having exon as well as intron with it. Okay, exon as well as intron is there with it. That is why this is called as hnRNA, hnRNA or heterogeneous nuclear RNA. It is having introns as well as exons. Okay, so such small things, the small terms in biology, we have to have a clear understanding. These small things will help you to solve the multiple choice questions which is coming in the NEET examination and such competitive examinations. So this channel is devoted for that too. So every single term is explained in a very simple manner. If you get any doubt in your mind, difference between what is this intron, exon, codon, cistron, gene, there are many terms related to. So these are the terms where students get a bit confusion and so they go wrong in the NEET examination. So this channel is devoted for clearing such things as well. So feel free to ask doubts if any regarding such things. Now coming back to our topic, heterogeneous nuclear RNA or hnRNA is having both exon as well as intron in them. So only when the introns are removed and the exons are joined like this, now we can call this as our mRNA. This is our mRNA. This is not now heterogeneous. This is having only exons. But here it was heterogeneous, having introns along with the exons. So now first reason why it happens in eukaryotes is clear, and how it happens is also clear to you, and we have come across the term hnRNA and mRNA, the difference. Now the next reason why it happens in eukaryotes, the next reason that I had written here is because eukaryotes have a nucleus. Prokaryotes don't have. Let me draw a rough structure of a cell here. This is the nucleus. So inside the nucleus we have this DNA and it has produced, imagine it has produced mRNA. Okay, mRNA. And now this mRNA has to come out of the nucleus. And as I told you, the process of translation or protein synthesis takes place in the cytoplasm. And my dear viewers, you know the site of protein synthesis? Exactly, it is ribosomes. Ribosomes are the site of protein synthesis, and you know ribosomes are present in the cytoplasm. Okay, and also in the rough endoplasmic reticulum they are present. So ribosomes are present in the cytoplasm. Fine. So this hnRNA has to come out with the information. It is there. It is carrying information from the DNA. It is carrying all the exons. It has got the information to produce the protein. Now it has to move towards the area, that is the machinery where this protein synthesis will take place. So that machinery is the ribosome, and that ribosome is present in the cytoplasm. Now this mRNA which is coming out of the cytoplasm, out of the nucleus, is a very new mRNA, nascent mRNA, and it is afraid of the outside world. You know, like when a small child goes to LKG classes for the first time, it is afraid, isn't it, to get into the classes and all. It cries for to see his parents and all. But once it gets accustomed, the child gets accustomed with the teachers or their friends and all, then the child is very happy to do the activities over there. Same is the case with our mRNA. This mRNA comes out of the nucleus, but it is very scared. So to protect this mRNA from the enzymatic actions which is present in the cytoplasm. In the cytoplasm, there can be many enzymes here. The cytoplasm, there can be many enzymes. There can be nucleases, ribonucleases can be there, which can just digest this mRNA which is coming out. So DNA is producing mRNA and mRNA is coming out and it gets digested. So whatever the cell has done will go in vain, is it not? So in order to prevent that, that means in order to safeguard this mRNA from the action of ribonucleases and to have a better part of protein synthesis to happen in the cytoplasm, two other complexities also takes place. That is capping and tailing. One cap and a tail is also added. It looks a bit, sounds a bit funny, isn't it? But it happens. A cap and a tail. So what is this cap and a tail? It's not like the cap and the tail that you are imagining now. These are all addition of some molecules. Towards the head region, you call it as cap. Towards the ends, 3' region, you call it as the tail. That's it. Okay. So why this capping and tailing? It is to prevent or it's to protect this mRNA which is coming out into the cytoplasm. So for protection this is done. So that is the next reason. The whole process of transcription is happening in the nucleus and then after it is getting formed, it is coming out into the cytoplasm. So in the cytoplasm there can be enzymes which can interrupt with the process. So in order to protect it in the cytoplasm, there are other two complexities. But then you will have a question in your mind. In the case of prokaryotes, they don't have this nucleus at all. They don't have this nucleus at all. And there is no hnRNA also. Isn't it? There is no hnRNA also. There is no hnRNA, there is no nucleus. Isn't it? So directly from DNA, mRNA is formed, there is no hnRNA and there is no nucleus also. Then why not these two processes happening in the case of prokaryotes? Are they protected on their own? It's not like that. In the case of prokaryotes, the process of transcription and translation are coupled. This is very important. In the case of prokaryotes, transcription and translation are coupled. That means as soon as mRNA is getting formed, along with that protein is also getting synthesized. It means both the process takes place one after the other. So there is no need of this protection. In the case of eukaryotes, mRNA is fully formed and after that it comes out and then only protein synthesis occurs. But in prokaryotes, as soon as mRNA is getting formed, along with that protein is also synthesized. So transcription and translation are coupled in the case of prokaryotes. Again, the question for your board exam question can be, why transcription and translation are coupled in prokaryotes? Why not in eukaryotes? Same two reasons it is. Reason 1, there is no split gene in prokaryotes. Reason 2, there is no nucleus in prokaryotes. So dear viewers, this particular 2 points that I have taught you is an answer for 2 questions. Why post-transcriptional modification occurs in eukaryotes? Or why transcription and translation are coupled in prokaryotes? For both these questions, answer is the same. Isn't that wonderful? Fine. Let's now focus on what is this capping and tailing. Let me first take what is this 5' capping. So as I told you, capping happens at the first end head part. You know, mRNA is produced in the 5' to 3' direction. So first let me write here as hnRNA. This is produced as hnRNA having exons, introns, and likewise. Okay, this is hnRNA. Now first step, that is under post-transcriptional modifications. Now we are going to learn the 3 steps. The first step is 5' capping. The importance of this step or the speciality of this step is that this is a co-transcriptional process. That means it happens along with transcription. It means this capping happens just after initiation of transcription. Just after transcription initiation, this step happens, that is 5' capping. So what is that? In the 5' end, so this is the 5' end, a molecule is getting added or a cap is getting added. And the name of that molecule is 7-methyl guanosine. 7-methyl guanosine is a molecule and that is getting added. You see here it is methyl guanosine. So this methylated guanosine cannot be recognized by any enzyme that is present in the cytoplasm. And so since the enzyme cannot recognize it, it will not go and cleave it out. It will not go and cut it out. It will not go and disturb it. So it is protected. Which part? The head part is protected by 7-methyl guanosine. Okay. Now you have the split genes as usual, exon, intron, likewise it is happening. Okay. Always it need not be exon, intron, it can be 2 exons together, likewise also it can be. Okay, just for you to understand I have written it like this. So it is 7-methyl guanosine cap is added. So first process is over and this is by a special linkage often asked for entrance, 5' triphosphate linkage. Okay, so explanation regarding this. So this process 5-methyl guanosine cap is not a simple process, it's a complex one. Explanation regarding each one will be, that is molecular basis of explanation will be given in the coming videos, in the coming sessions. Okay, so now just understand what are these post-transcriptional modifications, the steps, that's it. So 5' capping, it is done, 7-methyl guanosine cap is added and I hope you understood the purpose also for that. Now next step is the tail region, poly A tail. So there I hope you remember AAUAA sequence followed by UGU sequence, okay, where CSPT and all comes and cleaves. This sequence is cleaved. So this happens, I think you have learned in termination region. There I have taught you regarding this cleavage of this particular area and this region is now removed out. So this is how our mRNA is now. This is removed out and only this region is present. Okay, so let me connect these two. If I connect these two, it is like this. You have cap region here. Exons and introns are there. Exons and introns are there. And then AAUAA sequence is there. Okay, likewise, and there was a splicing in between them. Now this is our head region, it is already protected. Now tail region has to be protected. So this is done by the enzyme polyadenylate polymerase. Polyadenylate polymerase, in the name itself it is there. It will add many, poly means many, adenylate residues. Adenylate residues. That means many adenylate residues will be added like this. Many, 20, 30, 40, 200, 300 will be added here. Okay, many adenylate residues will be added in the 3' end. So that's how the tail region is getting protected from the cleavage action. So head region is protected, 5' methyl guanosine cap. Tail region is protected by poly A tail. And then comes, as I told you before, all the introns are removed. Cap is there, introns are removed, and exons are joined. Exons are joined here. And so now what you get here is our finished mRNA or proper mRNA. Now this mRNA can come out of the nucleus into the cytoplasm, and it is now ready for translation. Okay, I hope you understood the post-transcriptional modifications, the reason and why why it happens in eukaryotes and not in prokaryotes, and the 3 additional complexities. Often it is asked like this also. Which are the 3 additional complexities found in eukaryotic transcription? Why it is found? Why it is absent in prokaryotes? So I hope this session was really helpful for you to understand and to write those questions and answers. And dear students, if you have doubts any, please feel free to ask, and it will be better if you subscribe this channel and share it with your friends, relatives, near and dear ones, whoever needs this particular aspect of molecular biology. Go through the playlist, take the videos one by one, whichever is relevant for you. Please go through that, share it, and keep asking your doubts, and also share with me your comments. Waiting for all those things. Thank you all.